First of all, thank you for kind introduction, Professor Dr. Kim. Hello, everyone. My name is Okju Kim, CSO and co-founder of ATG Lifetech. First of all, I'd like to thank the organizer of JHBIC for inviting me to this webinar. Today, I'm very pleased to present under the title From Single Cells to Organoids, ATG's high-precision RNA-seq kit. In this talk, I will not only introduce our technologies, but also share how it can be directly applied in the field of microphysiological system. My hope is that this presentation will provide useful insight for those of you working on organoid chip, and other advanced preclinical models. Here is a brief overview of today's presentation. First, I will briefly introduce ATG Lifetech and our mission. Next, I will describe our core technologies, the foundation of our company. Then I will move on to our main focus today, ATG's high-precision RNA-seq kit. I'll explain why it was developed, how it improves on existing technologies, and how we validate its performance. Finally, I will present application case studies showing how our kit is being used in MPS research. To provide some context for our research, I'd like to start with a short introduction to our company, ATG Lifetech. ATG Lifetech was founded in early 2021 by three co-founders, Tae New, Yusin Jung, and myself. We shared a common background as early founding or team members of Cellemix, a Kostak-listed biotech company where we developed and commercialized various foundational technologies. This background gave us strong expertise in both technology development and translation to markets. ATG's mission is immune-based cell and gene therapies for a healthier future. To realize this, we have been developing three core technologies: single-cell multi-omics, genome writing and synthetic biology, and immune profiling. These technology development have continued with R&D funding from government bodies, including Samsung. Our ultimate goal is to leverage these core technologies to create innovative platforms that accelerate drug discovery and development. Let me now describe each of ATG's core technologies. Our first core technology is single-cell multi-omics. Obtaining high-quality genomic and transcriptomic data at the single-cell level is technically very challenging. It requires not only sophisticated protocols, but also substantial expertise in data analysis. Over the years, ATG has actively collaborated with universities, hospitals, and companies to refine single-cell workflow. We are now also engaging with global single-cell multi-omics device companies to expand this field further. One point I'd like to emphasize: at ATG, we operate the entire pipeline in-house, from library preparation to sequencing to downstream bioinformatics. We do everything internally. This ensures high data quality and which give us great control over consistency and performance. Also, we have an option of customized RNA-seq, and it is kind of user-friendly option. Let me share early results from our work in single-cell genomics. We developed a platform using laser isolation system for spatial genomics and cell capture technology to isolate circulating tumor cells from cancer patient blood with microfluidic chips. This allowed us to analyze the spatial genomics and genome of individual CTCs. To make a single-cell genomics more affordable, we also designed a barcoding system to lower costs and developed a method to analyze the genomes of fish-stained samples. These advances expanded the scope of what can be analyzed at the single-cell level. In transcriptomics, we conducted spatial transcriptomics on pre-labeled cells from cancer tissue sections. This approach helped us identify cancer stem cell biomarkers, demonstrating how spatially resolved single-cell data can reveal new biological insights. Building on this expertise, we are applying single-cell RNA to pharmacotranscriptomics. For example, we are collaborating with a Korean pharmaceutical company running a clinical trial in the United States for MASH treatment. Using RNA-seq to analyze patient samples, we aim to assess drug efficacy in the early clinical stage. However, as everyone knows, the obtainable sample from clinical trial patient is extremely limited, making it difficult to predict efficacy or clinical trial success. At the same time, regulatory shift means that animal testing is no longer mandatory in certain preclinical stages. As a result, organoid and organoid chip are emerging as new approach methodologies. ATG is actively working with both academia, such as Professor John at SNU, Professor Han at CMC, and Professor Kim at SNUH, and industry partners, including Acrobiosystems and company developing artificial platelet. To briefly introduce another core technology of ATG, nucleic acid, which are essential materials for gene therapy, inevitably accumulate synthesis errors during production. We are collaborating with Professor Che's group at GIST to develop error-free mRNA cancer vaccines, and this project has been supported by a 6-year R&D grant from Samsung. Lastly, we have developed NGS-based antibody discovery technology. By leveraging NGS analysis, we can verify whether page libraries are well constructed, track sequence enrichment across biopanning rounds, and using biochip-based platform, secure large number of high-affinity diverse clones. With that background, let me now focus on today's main topic, ATG's high-precision RNA-seq kit. RNA-seq has become indispensable for modern biology, yet results can vary significantly depending on the library preparation method. So selecting the right method for the objective is critical. As shown in the figure on the left, the RNA-seq profile differs depending on whether fragment occurs at the RNA stage or after cDNA synthesis. This choice alone can change coverage bias and downstream interpretations. Currently, the most widely used method is Illumina's TrueSeq mRNA stranded kit. It works well for bulk RNA-seq, but requires large amount of RNA, making it unsuitable for organoids or organ chip where RNA is limited. By contrast, SmartSeq2, originally designed for single-cell RNA-seq, can handle extremely small RNA inputs like 10 picograms. This makes it a good candidate for MPS applications. But here is the problem. Most public bulk RNA-seq data are TrueSeq based with advanced in single-cell transcriptomics like 10x Genomics' Chromium. Biomarkers previously undetectable by bulk RNA-seq can now be discovered by single-cell RNA-seq. Typically, biomarkers identified by single-cell analysis are then validated using bulk RNA-seq data. However, because discovery is SmartSeq2 based, while bulk RNA-seq validation is TrueSeq based, there is a growing issue that biomarkers found in via single-cell analysis fails to validate in bulk data set. So we asked, could SmartSeq2 be adapted for bulk RNA-seq? Although SmartSeq2 was developed for single-cell transcriptomics, what if we applied it as a bulk RNA-seq method? We tested whether SmartSeq2 is usable not only for single cells, but also bulk RNA-seq. We confirmed that the SmartSeq2 not only works for single cells, but also scales to bulk RNA-seq. As RNA input decreased, SmartSeq maintained similar gene expression. This is the Pearson correlation. So from the 1 microgram to 10 picograms, it shows the highly similar gene expression profiles. Also, regardless of the inputs, maintain the gene expression patterns, and also most NGS leads remained usable. Whereas TrueSeq did not like that. If the lower than 10 nanograms, there was no enough NGS lead in TrueSeq method. So in addition, as input decreased, the number of detected gene was higher with the SmartSeq2, and Venn diagrams Venn diagram analysis showed that genes detected by TrueSeq were almost always covered by SmartSeq2 as well. Next, I will describe what engineering was needed to optimize SmartSeq2 and which parameters were monitored to confirm performance improvements. SmartSeq2 employs template switching. When using MMLV type of RT reverse transcriptase, non-template addition of 3 cytosine is generated at the end of the cDNA synthesis. A template switching oligo TSO containing a complementary GGG sequence is then used to ligate produce cDNAs with the same adapt at both ends. Reverse transcriptase engineering and template switching oligo engineering and reverse reaction buffer composition have a major impact on template switching efficiency. These improvements collectively increase the template switching efficiency and overall kit performance. In addition, to improve overall RNA-seq library preparation performance, we performed TM5 engineering and buffer optimization required for NGS library preparation. Through this effort, ATG completed development of high-precision RNA-seq kit. So, which performance metrics should be compared? When benchmarking single-cell RNA-seq, 3 metrics are most important: cDNA yield and detected gene numbers and uniquely mapped lead ratio. As you can see on the right, for cDNA yield, if template switching efficiency declines, the template available for cDNA amplification decreases. Thus, cDNA yield is a primary indicator of kit performance. Next is the number of detected genes. In single-cell RNA-seq, the key question is how many genes are detected. Detecting as many genes as possible enables robust clustering and biomarker discovery to explain disease mechanism or drug MOA. Lastly, the uniquely mapped lead ratio. During the pre-processing of RNA-seq data, RNA-seq leads are aligned to the reference genome to generate gene count files. If the library fragments are too short for proper alignment, or if leads map to multiple loci, those NGS leads become unusable. By optimizing various conditions, our kit achieved over 200% improvement in cDNA yield at 30 picogram of RNA inputs, over 50% increase in detected genes, and higher mapping qualities. We have developed this improved performance product, are conducting performance testing with many researchers, and it is approaching official release. This demonstrated the robustness of our high-precision RNA-seq kit, which are preparing to launch soon. This section covers application of RNA-seq in the MPS field. Now, let me show how this kit is applied in MPS. Historically, drug development failure were largely due to efficacy and safety issues. More than 90% of drugs deemed safe in animals failed in human trials. This highlights the gap between animal models and human biology. This underscores the need for more human representative models at the preclinical stage. Recently, the FDA presented a roadmap to reduce animal testing. It is anticipated that within a few years, organoids and organ chip models will become the default in preclinical testing. Using ESCs or iPSCs, we can build organoids that mimic human organs. As a result, global pharmaceutical companies are interested in organoid applications, and diverse initiatives are on the way in the United States, Europe, China, and Korea too. Whereas past research mainly examined the potential of organoid models and their feasibility as animal testing alternatives. Going forward, we expect standardization studies aimed at obtaining consistent results regardless of country, institute, or operators. To achieve standardization, the first priority is to minimize batch-to-batch variation to secure batch consistency in organoid production. Many labs culture organoid inside the ECM domes, like on Madrigal. But it is widely recognized that organoid size and morphology vary within a dome. To address this, ECM-free methods such as microwave platforms or shaker cultures are increasingly being explored to maintain consistent size and morphology. Equally, the model reproducibility is crucial. When attempting to replace animal testing for toxicity evaluating using MPS, for example, with cardiac or liver models, functional outcomes for standard reference drug must be constantly reproduced. If beating period or IC50 vary across batches, the timeline for adopting MPS as an alternative to animal testing will likely be delayed. ATG is actively helping researchers improve MPS models. A representative example is our joint research with Acro Biosystems on organoids. To the best of our knowledge at present, Acro is the only company that sells organoid models to researchers globally. Conventional methods to access organoid quality include morphology size via 2D or 3D imaging, which has limit in measurable thickness, target protein expression with limited markers, RT-qPCR for small set of genes, and various model-specific characteristic assays. ATG has worked with domestic and international teams to introduce transcriptome-based analysis method for organoid and organ chip validation. What I would like to emphasize is that ATG's RNA-seq has no practical RNA input limitations, enabling us to monitor differentiations over time, even at the single organoid level, and to confirm at the endpoint whether gene expression profile are consistent across batches of a single organoid or organ chips at the transcriptomic level. For reference, although single RNA-seq is often used to advance MPS models, it is primarily used to check how well the model differentiates and how diverse the cell types are. Let me share a case study on batch consistency in organoid production with Acro Biosystems. As a company that sells organoid models worldwide, Acro aims to supply organoid products of consistent quality. In addition to QC by size measurement, IF imaging, and RT-qPCR, Acro introduced transcriptome analysis with ATG to evaluate how uniformly organoid models express gene across batches. For the experimental design, we prepared 10 organoids per batch, 30 organoids total across 3 batches. Of these, 2 organoids per batch were fixed and returned to Acro Biosystems in United States for IF imaging, and 8 per batch underwent RNA extraction and RNA-seq at ATG. To comparatively assess how uniform gene expression was among organoids, we used 3 healthy donor heart tissues and 5 PBMC samples as controls. After generating RNA-seq data, we accessed clustering via principal component analysis (PCA). Regardless of production batches, organoid clustered together, indicating high similarity in gene expression patterns both between and within batches. One point to note is the difference from healthy adult heart tissues compared to cardiac organoid. It is well known that most organoid models exhibit lower maturity, showing gene expression patterns close to fetal rather than adult tissue. So our results show the same with clear difference from adult heart controls. We then applied the permutation test to assess the similarity among organoids. We performed 1,000 randomization shuffling organoid samples to evaluate whether batches still shows no gene expression differences when converted to distance. For reference, the top panel distance represent technical duplicates, showing that ATG's RNA-seq kit experimental variance is minimized. On the right, The permutation test shows only very small, only a small increase in distance among organoids, whereas PBMCs, which naturally shows larger individual differences, exhibit much larger distance variance. Comparing between organoid batches, distance changes were small, with a p-value of 0.18 to 0.72, indicating no significant difference between batch groups. In contrast, comparison between organoids and tissues and PBMCs showed less than 0.001 p-values, indicating clear differences. This demonstrates that ATG's RNA-seq kit provide a reliable way to measure and ensure organoid batch consistency. We also analyzed the heat map using public database. Selecting heart-related genes, we observed that expression in cardiac organoids and heart tissues were largely similar. This red line is the organoid samples, and the others is the tissue and public tissue database, heart tissue database. So heart tissue and cardiac organoids shows the highly similarities, while the being clearly distinct from other organs. Our second example is a gut, liver, kidney multi-organ chip used to model pharmacokinetics of diclofenac. Many experts in the MPS field believe that while MPS can replace the animal models for efficacy and toxicity testing, replacing PK models used for pharmacokinetic studies will be more challenging in the near term. This project involved triple multi-organ chips to model drug PK using diclofenac, a well-studied NSAID. We accurately predicted metabolism and distribution in the multi-organ chip. However, there was a limitation. We could not be sure that each organ chip's drug response matched clinical outcomes. Therefore, together with Professor Han at CMC, ATG applied RNA-seq in parallel with PK modeling to verify whether each organ chip's response aligned with clinical result. RNA-seq results show that post-treatment expression change in gut on a chip were associated with the inflammation and COX inhibition, consistent with diclofenac's well-known GI toxicity. We expected that incorporating RNA-seq into multi-organ chip PK models so that each organ chip's transcriptomic response can be checked against the clinical outcomes will enable the development of more advanced models. Our third case study involves tumor microenvironment chips. We are conducting joint research with Professor John at SANU to build a biochip that models the TME for evaluating the efficacy of new drug candidates. Accurate modeling of the TME is crucial for assessing drug efficacy and mechanism. Typically, when testing drug on TME chips, only staining-based phenotype are examined. By adding RNA-seq, we can observe how cancer cells respond to drug at the gene expression levels and also identify mechanism of action. In this study, we examined which gene pathways were activated depending on tumor mimicking cancer cell size and drug type. Our fourth case examines time series RNA-seq on the angiogenesis chip to track transcriptional dynamics. Typically, organ-on-chip readout stop at phenotype or handful of protein markers, which limits what we can infer mechanistically. In this study, we profiled the angiogenesis chip from day 1 to 6 and applied a likely ratio test to the time axis to extract the signature genes that change significantly over time. Using those temporally regulated genes as input to KEGG enrichment, we identified the pathway linked to angiogenesis and autophagy. While recent reports suggest a connection between autophagy and angiogenesis, our results show that temporal transcriptome approach can find this relationship directly from the chip rather than relying only on endpoint phenotypes. The broad point is that the same concept can be applied to other organ chips. Instead of just confirming measurable phenotypes, time-reserved RNA-seq reveals previously unappreciated gene programs, enabling mechanism interpretation and biomarker discovery that improve model insight and translatability. At ATG, we are committed to developing core technologies that support drug discovery and development. These technologies have been validated through publication in leading journals, and we are now focused on applying them in real-world collaboration with academia and industry. By combining advanced RNA-seq with MPS models, we hope to accelerate the transition toward more predictive, reproducible, and human-relevant preclinical systems. Let me wrap up with a summary. Based on our single-cell multi-omics platform, ATG developed optimized RNA-seq library preparation method tailored for organoid and organoid chip samples. With this method, we were able to confirm the batch-to-batch uniformity of organoid production in collaboration with our partner, Acrobiosystems, by directly developing the reagent used in RNA-seq. We give researchers the freedom to customize their workflow, ensuring that they can apply the most appropriate technology for their specific course and consistently generate high-quality, reliable data. Ultimately, these efforts are not only advancing organoid and organoid chip models, model validation, and supporting drug discovery, but they are also creating high-quality transcriptomic data set that can be applied to the development of AI models in biomedical research. Yeah, this is end of my presentation. Thank you for your attention.